Hello everyone. Welcome to today's live broadcast by Dr. John Iafrate. I'm Dr. Jared Snyder, Creative Marketing Manager at Archer DX, and I'll be your moderator today. Here's how this presentation works. We want to hear from you. Questions, comments, and even answers can be submitted via the green Q&A button at the lower left of your screen. We'll try to get to everyone, but if we run out of time, we'll make sure to follow up with you by email. You can enlarge the slide window by clicking on the screen icon in the lower right hand of your slide window. If you can't hear or see this presentation properly, let us know by clicking on the support button at the top right or the Q&A button in your lower left. We'll monitor both. Now let me tell you a little bit about Dr. Iafrate. He earned his MD and PhD from State University of New York at Stony Brook School of Medicine and went on to Brigham and Women's Hospital for his residency and fellowship. He's currently the medical director of the Center for Integrated Diagnostics at Massachusetts General Hospital. John is an innovator who identifies problems with the status quo and works to solve them. As a molecular pathologist, he recognized shortcomings with the current methods of mutation detection. And so over the last 7 or 8 years, he and his colleagues have developed technologies that enable large-scale molecular detection and to get more information from less input material in a shorter amount of time. And with that, I'll turn the time over to Dr. Iafrate.
Okay, thank you, Jared. Welcome, everyone. I appreciate you guys signing on and listening to this presentation. I'm going to cover today some technology developed in our laboratory here largely by 2 very talented scientists, Dr. Longley and Dr. Zhongli Zhang. I get the privilege of presenting, but they did a lot of the presentation, and I just want to give them some upfront credit. The technology we developed, called anchored multiplex PCR, I'm sure most of you have heard about it if you've been looking at the Archer website and some of our publications. We really developed this early on as a method to move a lot of our fusion detection analysis that we were using either FISH or IHC into the next-gen sequencing world to get more cost-effective and higher throughput. So the AMP technology really was a fusion-based technology, but we've begun to mine it and improve it and move it into other areas like SNV detection, CNV detection, and RNA expression. So that's what I'm going to tell you about today. Obviously NGS in cancer analysis is a very, very big field. We have about 1 hour to go through the material, and so this is not going to be a comprehensive overview of next-gen sequencing. I'm going to give you a little bit of clinical background and then really focus in on the AMP technology. Obviously, our team is biased on it. We've developed the technology and we think there's a lot of advantages. I will touch on where it compares to other techniques, but it's not going to be an exhaustive review of other techniques which obviously have their place and have shown great promise in the area of tumor genotyping. So outline practical challenges in clinical genotyping, just a couple of slides just to put everyone on the same page about clinical genotyping. Then we're going to talk specifically for most of the talk about AMP technology, fusion, SNV, CNV, RNA expression, and then a little bit of clinical scenarios to show you where we're applying and how we're applying the technology at Mass General Hospital. Obviously, that's unique to our own. So before I start, I just wanted to give a little bit of a conflict of interest disclosure. The talk is sponsored by Archer DX, of which I, along with Long, Zhongli, and Jason Myers, were founders. I continue to be an SAB member and an equity holder in Archer. With that said, I do want to emphasize that today's talk is my own content. The data I'm showing you is based on the MGH AMP research. I'm not really going to be talking about Archer kits or Archer specific tools today. For those, please follow up with Jared and the Archer team after. The talk was not approved by Archer DX, so it's my own content. And so that's probably good or bad. If you're wanting to get more specifics on the Archer products, I'm not going to talk a whole lot about those specifically. On the other hand, I would add that we are obviously in close contact with the Archer DX team, and a lot of the tools that we've developed, I think in parallel, they've moved to commercialize. So a lot of the things that I'll be talking about, I think, will be very applicable to any commercial development and an interest in your particular lab on bringing on the technology. Okay, so with those conflicts disclosed, I'm gonna, yeah, as I said, give a little bit of background for a couple of slides just to put us all on the same page. Most of my interest in cancer genotyping started or really accelerated around the development of the drug crizotinib. I run the molecular diagnostics group at Mass General Hospital, and we had been quite busy over the years. Give me 1 second. I have a little pen here that I'm going to see if I can use. There we go. So we had started early on in the development of crizotinib, actually before crizotinib was an open clinical trial. We were involved in clinical pathologic analysis of ALK-positive cases. Dr. Mano's group from Japan first described this in 2007. We started screening these for ALK fusions in lung cancer just a few months after that paper was published. And we used FISH. We did many FISH tests, and in fact became the central lab for the phase 1 trial of crizotinib, and got to play, I would say, an integral role, at least early on the development of companion diagnostics in that space. So we learned a lot about companion diagnostics and how important it is, even for a relatively rare genotype, to uncover actionable, targetable mutations. So happy to talk offline with anyone about our experience there. I would say the most exciting thing and kind of the lessons we learned about crizotinib is if you do it right, if you have a good target and you have a good drug, you can move things along quite quickly. So Dr. Mano's group, as you know, discovered EML4-ALK in 2007, and then accelerated approval of crizotinib was obtained in 2011. About as fast as you can go from what is a new drug and a new indication to some FDA approval. And so a lot of what we've done afterwards is trying to match this rate, and this is a hard rate to keep up with, but I think something that everyone in the field, probably a lot of you on this call, are excited about in your particular tumor type. Regulatory challenges. For those of you who don't spend your time thinking about clinical genotyping, I'd like to give a couple of slides to put us all on the same page. There's a number of entities that control what we do, at least in the United States. The FDA wants to know, does your device work? Obviously, genotyping is considered a device, so they want to know about clinical and technical validity. CMS is important. Should we get paid? Is there clinical utility to what we're doing? I think this is our major hurdle for our field of next-gen sequencing or clinical genotyping. The AMA provides codes. We'll talk about that in a second. And then CLIA. Many of you probably have heard about CLIA that don't run CLIA labs. CLIA is not asking, does the device work necessarily the same way the FDA does. The CLIA regulations really want to ask, does the lab know how to use the device? Do you have the proper quality assurance and quality control? So any recent developments? In fact, there have been a lot of recent developments around the FDA and the FDA regulation of tests. For labs like ours, academic labs, there's been a lot of interest and publication of draft guidelines around the regulation of lab-developed tests. By lab-developed tests, that means tests that are not FDA approved. And in the cancer NGS space, I could ask the question to everyone on the team on the call: how many FDA-approved cancer NGS tests are there right now? And the answer is easy. There aren't any panel tests yet approved. So the space we're working in for now, obviously some of the big players, including Illumina, Life, Archer, QIAGEN, are going to be very interested and are very interested in proceeding down the road of getting FDA approval, and that we'll see in a few years. CMS, as we talked about, is a big question. Should we get paid, and can we get paid? I would say I'm also happy to talk to anyone offline about this. We spend a lot of time right now talking to payers, whether private or public, Medicare, Medicaid, to see, can we come up with reasonable payment schedules for NGS. The AMA has published 3 codes for NGS, the 81445, 81450, and 81455 in the US just this year, which provides us the ability to bill, but it has not yet, I think, reached its full potential, and that we really don't have payment rules and guidelines published from each of the payers. So we still have some work to do. So that's currently where we stand. I'm going to come back to this timeline slide just for one thing. I do like to talk about LDTs and the importance of lab-developed tests and progress in the area of molecular diagnostics. In my opinion, it was the early launch of our LDT ALK assay that allowed the rapid approval of crizotinib, the rapid progress of crizotinib. We wouldn't have been able to launch that test as a clinical test if it wasn't for the ability to launch LDTs. And in fact, I think the approval of crizotinib would have been delayed at least 2 years if we had not been allowed, or if the regulations surrounding LDTs had been more stringent at the time. So the next slide is an important slide for those of you who run labs. Many of you are well aware of these terms, but when we start talking about an NGS platform and why we developed AMP, you can look at some of these considerations. For a clinical test, we need it to work from archived FFPE material. We need analytical sensitivity, especially for SNVs, down to 5% or lower. Our clinicians put a lot of pressure on us to get less than a 2-week turnaround time. With NGS, it can be a challenge to get the library construction, bioinformatics, and reporting out within 2 weeks. And so those are the major considerations why we started looking at developing our own technology, especially around fusion detection. Actionable targets, this is an area of much negotiation, much heartache, I would say. Everyone has their own list of actionable targets. I'm not going to spend too much time, other than our philosophy is we should be as comprehensive as we think we need to be, where everything on that panel is actionable. So there's a happy medium between the number of targets, the amount of sequencing someone, I think, should do, and the cost for doing that. And so the happy medium there is picking highly actionable targets. And this is not really a talk about clinical utility and sustainability. However, the elephant in the room, I think, for any talk like this is nothing that we're talking about is sustainable unless we have some clinical utility proof. We're involved in the NCI match trial, which is one of those type approaches, a large multi-center, actually multiple cooperative group, NIH-funded study to do such a thing, and we're excited about we're excited about trials like that. But I would say we're still a few years away from being able to show foolproof evidence of clinical utility for next-gen sequencing. 2008, we launched SNAPSHOT, or Original Genotyping Assay. It's a hotspot mutation detection assay. It served us very well. If you look at the gene list and the mutation, it really has almost, not almost all, but many of the important recurrent targetable genetic alterations. And it still is it's a quite powerful assay that can be done cheaply and quickly. So in order to move on, we really, to the next technology, we really need something that would compete favorably with something that could be done very cheaply and quickly. And so that's why we started developing the in-house AMP technology long and long we did. Profiling in the SNAPSHOT era, this is lung adenocarcinoma. One thing that should pop out right away is there's no mutations that we would found in this type of panel, about 40%, and to us that was showing that we're not helping as many patients as we could have. So we obviously had to move to NGS. And then tumor suppressors like P53, when you look at a hotspot mutation assay, you just don't get good coverage of tumor suppressors because you are looking at hotspots. So for those 2 reasons, we moved on to NGS, and I won't belabor that discussion. Clinical utility, I did mention, this is my last intro slide, clinical utility. There's not been a lot of papers out there showing the usefulness of next-gen sequencing or broad genotyping for that matter. This was a paper from 2012 from the MD Anderson group. And I thought it was, it was not a randomized control trial, but it was a very interesting analysis of their phase 1 clinical trial data where they genotyped many, many patients using a similar assay as our original SNAPSHOT, and show the response rate is much higher in patients who are genotyped and go into a genotype-directed trial than other phase 1 patients who don't. And so that type of data is out there, and we're beginning to see more of that, and that's going to be very important for payers. Okay, so that's the end of the introduction. Let's move into NGS proper. What are we doing at MGH in 2015 right now? If just to let you know how we view it and what we're most interested in doing, we prefer medium-sized, flexible panels. The need, I think our team all feels, is to balance sequence costs and comprehensiveness. Again, actionability is important, and for that reason, we're currently working on five panels. Solid tumor DNA for SNVs, indels, and CNVs, solid tumor fusion, sarcoma fusion, hematologic tumor DNA, and separate heme fusion. And by separating them out into different panels, you accomplish a few things. There are less genes per panel, and you can run more samples per run, per HiSeq or MiSeq or PGM, whatever you're using, and obviously keep costs at a minimum. So to us, this balances those two things of cost and comprehensiveness. The list may look similar to what Archer DX provides, and I would say we've worked together with the team there that we're very close with to look at the gene lists and make sure the content is somewhat in line. So some of the things I'll be talking about, while not exactly Archer kits, will be very similar to what you would find from an Archer offering. The technology, I have a couple of slides describing the technology. I think this is very important to go into the details of why AMP is very interesting. Zhongli and Long worked very hard to develop this. It ends up being, I think, quite a simple approach, and I think quite elegant in its simplicity. We're talking, to start with, we'll talk about translocations, but the same workflow, the same primer design basically can be used for genomic DNA. There's really no difference other than your starting material. Whether you start with double-strand cDNA, which is how we start with our RNA sequencing or translocation detection, followed by end repair and A-tailing, and then using these adapters. They're Y adapters, hemifunctional Y adapters, where only one of the Ys is functional, then gets ligated. So both sides of your library, this is a random library of double-strand cDNA, gets your adapters ligated, so it's on both sides of your fragment. And then you do 2 rounds of PCR, where one of your primers is within the adapter tail, and the other is a gene-specific primer, where your gene or region of interest is just upstream of your GSP1. A lot of work that Long and Zhongli did showed that doing 2 rounds of PCR was very, very important to get high on-target rates. And so using the GSP2, a second round of PCR gives you over 90% on-target rate. And so the assay, as you can see, is largely a PCR-based assay, but it has a couple of interesting components, which is this upfront random ligation event. So it has a little bit of hybrid capture, random end type sequencing we'll talk about, combined with some of the advantages of PCR, which is it works quite well with low input material down to 5 to 10 nanograms of input material. If you were to use genomic DNA, instead of having randomly produced double-strand cDNA, you would have we would shear double-strand genomic DNA, and that would go into the library. I'm just going to give you a brief introduction in this slide. We'll go into more detail about what the sequencing itself looks like. There's some real advantages to the AMP technology. It has a very unique appearance when you look at a sequencing pileup. Whole genome sequence, hybrid capture, has this Gaussian distribution of reads where your capture probe would be in the middle, and you're capturing different fragment lengths around that area of capture. And you have multiple start and stop sites, and those can be very useful for a variety of reasons, quality control, and looking at the diversity of your library. It's very difficult to analyze diversity with PCR-based, amplicon-based sequencing. So many of the clinical application platforms for cancer, where you need to go down to very low levels of input material, use PCR-based primers where you get identical start and stop sites. It's very difficult to assess complexity of libraries when all of your reads start and stop at the same spot. For the AMP technology, we have actually 2 primers for each target. Again, you have random ends because of your ligation of your adapters is random on either one of your ends, and then we actually come with a primer on the plus strand and a primer in the minus strand, and so that actually gives you 2 different distinct read pileups that you can track for each particular target. And since you have random ends for those reads, you can actually track a number of things. You can do error correction using molecular indexing. Standard deduplication techniques work just fine as well. And you can assess the diversity of your library, which is incredibly important for low input FFP materials that we'll talk about. So I'm going to talk about two things on this slide that I think are an advantage that we've tried to incorporate into the technology. Can be incorporated obviously into other sequencing technologies. I'm not saying it's unique, but we've tried hard to make use of molecular indexing and bi-template sequencing to reduce the false positive rate. So if you remember, you have these two Y adapters on your fragment here. So let's say this is a genomic DNA fragment. This dotted box is an exon of interest. We actually have a set of plus primers and a set of minus primers. And so these two can behave together, behave very well together in the same tube, and allow us to generate two sets, let's say overlapping sets of library fragments. Off the plus primer, you can see the anchored side is this side has a flat end, and then there's staggered start sites on the opposite end as you extend randomly to these to where the shearing sites are. We have these ends on the in the adapter, which are molecular indexing and allow us to bioinformatically group identical original template molecules, if that makes sense. And we'll talk about why that's important in a second. So for the plus strand primer, we actually get a set of reads. I'm not showing it here, but you have both forward and reverse coverage on a sequencing read. And then the minus primer, you have the opposite. So you have anchored side here and then staggered sides there. The minus strand, you have forward and reverse as well. The error correction comes in, it's very interesting as you can separate the plus and minus. You can actually correct for things like C to T mutations that are common in FFP tissue. So in this case, I'm showing a C to T mutation here only on the plus strand, but not on the minus strand. So that off the bat tells you right away that there's an artifact here. And the C to T would only be happening, the oxidative deamination would only be happening on one of the two strands because you'd have a G on the other strand. So you'd only see a mutation in one but not the other. And that allows us to eliminate a number of these C to T mutations that are quite challenging in FFP material. There's another aspect here, which is the molecular barcoding, molecular indexing, or standard deduplication that is really important. So if you look at these reads right here, these top four I've actually designed on purpose to have the exact same length. Let's say these were the original same template molecule. They'd share the same the same molecular index, exactly index, and bioinformatically they can be grouped. And if we have a T mutation in a subset of those reads, not all of them, not a consensus reads, we would rule that out as a false positive. So the molecular indexing and deduplication abilities allow us to make these bins that we can do intra-bin correction that lets us get rid of these false positives. And then the separate plus and minus strand by template sequencing allows us to do that. A real mutation would be present as shown here as A's on both the plus and minus strand. And again, a real mutation, if it was present within this particular original template molecule, would be present within all the identical PCR duplicates, right? So you can see, we'd see all four have the A there. So I would say we're just beginning to really optimize how to utilize this type of error correction and mine this kind of data. So we're excited about it, and we think the ability to work with low input materials and gain some insight into the error rate is really important. So almost all the assays we do, this was designed on purpose by Long and Zhongli, utilizes FFP material, and we extract total nucleic acid. So we simultaneously extract RNA and DNA into a single tube, and that can be utilized for all of our downstream applications, which we currently have. These are actual screenshots of data that we have using AMP for RNA expression. This is a 50 gene profile for RNA expression. RNA fusions showing a CD74 NRG1 fusion, DNA copy number assessment, which is relatively straightforward, as well as SNVs and indels. So single starting material, we can get almost everything we want, and this can be done out of low input material, so we're pretty excited. Let's talk for a few minutes about the challenges around RNA, these different assays. RNA expression, what are the challenges? As everyone knows that's done this over the years, normalization of data is not straightforward. For NGS-based, especially targeted RNA expression, balancing of primers, we'll talk about, is really important. There's major challenges around tissue-specific expression, especially in the diagnostic arena. AMP offers several advantages. The design is easy. You have the ability, to boost primers when you need to, and all this can be done relatively easily and in a customizable way that's fast. RNA fusions, what are the challenges? There's a lot of challenges around bioinformatics of calling fusions, mapping to basically split reads and making sure that you can find both partners. Off-target effects, the actual mapping of sequences can give you off-target effects that are dependent on the expression level of an individual target. So you may see an off-target effect only at in a sample that has high-level expression of a particular RNA. And then we'll talk for a slide or two about limited detection testing, which is really challenging in the clinical and clinical trial arena. The advantages of AMP: you need only one primer, easy design. DNA copy number challenges, normalization of data. Low copy number cases remain a challenge. The advantages of AMP: molecular barcoding. SNV and indels challenges, mutations under primer. Since it's somewhat of a primer-based, PCR-based assay, we still have some of the issues around mutations under primers. However, the bidirectional coverage allows us to get around that. The data format of AMP is unique, and it's taken us some bioinformatics work. Some of the standard platforms out there for mutation calling are not optimized for AMP type data, so we've had to work on that. The advantage of AMP, again, are staggered star-site error correction and bidirectional coverage. Let's talk for a few minutes about low input materials. And I think this is very important as you compare something like AMP, which will give you good data at 5 nanograms, to an amplicon-based, a pure amplicon-based assay that has neither molecular indexing incorporated or the ability to assess complexity. So if you take a sample and you put into, this is an AMP-based DNA assay showing read coverage over an EGFR exon from 200 nanograms to 5 nanograms, what do you see? Well, up at 200 nanograms, you see an even distribution of read start and stop sites. And so that shows you a very high quality library. While it's down at 5 nanograms, you may have a lot of reads. You may have, let's say, 10,000 reads up here at 200 nanograms. You can still have 10,000 reads down here. But you're beginning to see this very blocky read pileup, where these blocks represent large numbers of identical reads that have been PCR'd over and over, and they give you this kind of step function. So when we see this visually, we know this is a low complexity sample versus this smooth distribution is a beautiful high complexity sample. Why does that matter? That matters because while you have 10,000 reads down here, you actually probably only have 10 or 15 unique reads. And so if you're quoting your sensitivity based on a validation run done at 200 nanogram sample, where you can detect 1 in 100 or 3 in 100 mutant reads here, that sensitivity does not relate, is really not relevant to a sample down here at 5 nanograms, where you only had 20 reads to begin with. So the best you could ever do is 1 in 20. And if you'd like 3 reads, your best sensitivity ever would be 3 in 20 reads. So you have a much lower sensitivity down here, something you really can't tell from standard amplicon sequencing. We see this also not just in low input, but in poorly preserved samples, older samples, and so getting an idea of the complexity of the library is incredibly important in the clinic. This is giving you another idea. This is read pileup over 4 exons in the KRAS gene. This is data Zhongli had provided, which is showing you in turquoise the raw data coverage, that is the raw reads, the non-consolidated, not using the molecular indexing to consolidate read duplicates. But once you consolidate them in this black peak, you can see the percentage of actual total reads that are unique can be quite low in an FFP material. So this could probably be about 10% of the reads are actually unique. And that's a huge difference, and it's incredibly important in thinking about sensitivity in cancer genotyping. If you don't really need to see the detail of this. This is a slide pileup using our custom viewer that Long has developed. It's a pileup around one gene. I'm showing you just the plus reads up here, the minus reads down here. Let's ignore those. You can see this pileup over this particular gene. I don't remember which gene it is very blocky. So you have many nucleotides here. You probably have 20 nucleotides between here and here, but you have no reads that start or stop there. And so this is a very low complexity sample. And if you don't remove duplicates, what do you see? Well, let's say we have in this pileup 200 reads. We have 2 variant calls here that look real. a red one and a purple one. The red one, these are probably about 10 to 20 reads in that little clump here and 10 to 20 reads in that clump. And interestingly, if you look out to the ends, you can see these clumps are actually all start and stop at the exact same place. So while we had, let's say, 40 reads out of 200 showed this mutation, we actually know there are only 2 mutant reads. And that's a very different, sensitivity and a very different level of confidence in the clinical lab, knowing you only have 2 reads. And here in purple, we likely also only have 2 reads, even though probably 20 to 30% of the total reads in this call were mutant. It really is probably just 1 or 2 original template molecules. So when we see stuff like this, we get very concerned and are very careful in our interpretation of the data. So that's a general background into how AMP works and how we utilized some of the error correction and complexity information. What are we actually doing? We have a snapshot replacement panel. We call it NGS, snapshot NGS, which is also AMP-based. We have about, actually, I'm sorry, I should have updated, it's about 50 targets now. Many of the original snapshot genes are there, except we now have full coverage of some of our tumor suppressor genes like P53, SMAD4, STK11. And so we have good coverage with all of the advantages that we were talking about. And we have about a 2% to 5% analytical sensitivity, and we think we can get things done quickly, with a reasonable price, with a small amount of input material, which is incredibly important for a clinical lab, which really deals with many, many very small samples. We've gone from 60% of our lung adenocarcinomas to have detectable drivers to over 85% of them having detectable drivers. Some of the other specific issues that I'm going to talk about with SNVs and indels are we do deduplication as looking at the molecular indexing, as well as standard deduplication followed by mutex variant calling for SNVs. Longseam has developed a custom indel caller that we use, taking advantage of bidirectional coverage assessment. The analytical sensitivity that we found in our validation studies are less than 5% across the board for all of our validation samples that we use. We had a lot of older snapshot samples that we could use, and so the assay performed quite well on genomic DNA. Just to give you some novel stuff, if you've heard some of these stories before, we've actually begun to uncover, although this is not an ultra-sensitive technology, this is not necessarily something that you would use for monitoring solid tumors in blood or plasma, it is quite sensitive. So for many, we feel comfortable down in the 1% range if we have enough coverage. And we've begun to find, I would say, unexpected heterogeneity of common drivers. So I'm going to show you some examples, recent examples that we found of NRAS and IDH1 mutations in AML and MDS, which are really quite interesting stories. These are put together by Valentina Nardi, a pathologist in our group, so I give her some credit here. IDH1 and 2 is of very high interest, obviously, to us in the diagnostic lab, as well as to some of the therapeutics clinical trial at pharmaceutical companies. What I'm showing you here are 5 patients that we've performed genotyping in 5 patients with AML that we performed genotyping in. In gray are the blast counts, so it's presumably the tumor burden, and then in blue and red are IDH1 and IDH2 mutations that we found in the same patient, that is within the same patient's tumor. I had always assumed, and I think it was a general assumption by many, maybe not all, people in the field, in the AML field, that IDH was an initiating event. We have some cases like patient 3, where clearly the blast count is much, much higher, even if you add the 2 IDH1 and 2 components together, those 2 subpopulations together. So these patients, which have both IDH1 and 2 mutations, really support the idea that there's an initiating clone on which background there's selection for mutation in the IDH pathway. So you may ask, well, are these occurring in the same patient or not? This is a case where we actually have 2 separate IDH2 mutations in the same patient, and what you can see from the read pile up here is that the mutations are actually in different, are in trans. They're on different alleles. We've not been able to prove yet they're in different cells, but they certainly are in different alleles. And so that interest has led us to look at some other genes, and we found actually in AML as well that there's common heterogeneity in the NRAS gene, obviously a well-known common driver, and the mutations are in with the hotspot codon 12 and 13 loci. So this is a case that we found recently that had four non-overlapping mutations. I can show you the read pileup down here. I'm only showing you a small portion. There's probably another couple of hundred reads supporting. But you can see the main mutation is there, but there's also another mutation here, another mutation here, and there's two here. And interestingly, if you zoomed into it, which I don't have, they're all mutually exclusive. So this is something we've begun to see in a number of cases, and this is a second case from a few months ago, also Valentina's case, that had 3 NRAS mutations. This is a patient with myelodysplastic syndrome, and you can see 1, 2, 3 separate NRAS mutations, and all mutually exclusive. So we're quite excited. Although this is not a discovery exome platform, we are excited that its very good sensitivity is allowing us. Well, one, it allows us 2 things. It allows us, one, to have a very sensitive assay that works well on low input material samples, so we don't reject that many samples in the clinical lab. And number 2, it's beginning to reveal some heterogeneity, which is exciting, but I would say probably more stressful than exciting right now for the clinicians who have to deal with this. SNV applications, I'm not going to talk about the general applications all of you know about, EGFR testing, BRAF testing. I'm not going to go into those kind of details, which I assume everyone is well aware of. But some of the new applications we're looking at that I'm most excited about are in cytologic samples. Many of you probably know about Yuri Nikoforov's work in thyroid from the University of Pittsburgh, done some great work. I think following on that, we were excited to apply NGS, and we've applied our solid tumor panel to several cytologic samples, including pancreatic cysts, which we basically standard of care at MGH, that all pancreatic cyst fluids get sent for NGS. And this study, which is nearly completed with Martha Pittman in our cytology division around bile duct cytology, which can be very, very difficult. So in this study, we used our NGS approach, the DNA approach, to take cytological samples from bile duct scrapings, biopsies. These bile ducts are within the pancreatic head, or the common bile duct, or extrahepatic, the extrahepatic bile ducts, or the pancreatic duct, sorry. And in this particular study, we looked at about 74 patients. 81 total cases. We got material, we extracted the DNA, and then we performed genotyping on them. As you can see, of those patients, 21 had KRAS mutations. Interestingly, 14 had P53 mutations. And so what does all that mean? Well, we had clinical follow-up on them, and we use clinical follow-up as kind of the gold standard for determining sensitivity and specificity of these assays. Cytology, as any of you in pathology know, is not great at bile duct. From a sensitivity standpoint, for those patients who ended up with a positive clinical follow-up for neoplasia or malignancy, cytology was only sensitive for 67%. FISH, which is commonly used as an adjunct, was sensitive in 55%, and NGS in 74%. So NGS, at least in our data set, was better. And when you compared, when you added cytology and NGS, you got a boost to 85% sensitivity with a very, very good specificity. So a study that was a fair amount of work, but I think very exciting, that showing NGS can be really important, not just in predicting which drugs patients should go on, but can help pathologists make diagnoses. So in the diagnostic arena, similar to, I think, how Yuri has looked at some of his thyroid work. Interestingly, maybe not surprising to some of you, P53 mutation was correlating not just with neoplasia, but with malignancy. So 13 of these 14 P53 mutations at follow-up had a bona fide diagnosis of malignancy. And we don't have a whole lot of markers, molecular markers, that would allow you to say that is not just a neoplasm, like KRAS can tell you it's a neoplasm, it can't tell you it's malignant. P53 mutation in this setting is quite strong predictor of this is a malignancy and we got to do something aggressive. So we're excited about that. CNVs, we look at CNVs, and commonly every case that we run through our genomic DNA assay, our solid tumor assay, gets CNV coverage. It's a simple, we do this simply by looking at the read coverage. In this case, we're looking at several exons and introns. 5 base pairs into the introns allows us to do read counts at those positions. Obviously, you would not be getting RNA interference for reads that extend into the intron. So almost all of our GSPs are within exons, and if those read into introns, we can just read those and take the median of those over particular genes and then normalize that data, and that gives us our CNV coverage. We've been happy with it. I'll spend a few minutes talking about CNVs. There's obviously a huge amount of interest. NGS, along with RACGH and other PCR, other grind up and analyze assays, as I call them, they're very good at detecting high-level amplification. So this is a GBM, which a lot of people use because they have so much EGFR. In this case, is an EGFR amplification that AMP clearly picked up very easily. You see the read count is way off the top of this log2 scale. And in this case, we actually confirmed by FISH. We currently are confirming all of our amplicons by FISH. In this case, it confirmed. This case was interesting because it actually had a focal, in this case you can see over here, a focal PDGFR alpha amplification here. There's about 3 or 4 cells there, but it was still picked up by AMP. And then several other genes were ROS and MET were normal, and we had FISH confirmation of that. So we're happy with the performance. Again, why are we excited about it? Most platforms these days have fairly robust CNV detection. Low input material, low input FFP material seems to be performing really well. So for summaries of our CNV work, what can I tell the audience? I can say we confirm all potential amplifications with FISH. We don't have a hard cutoff in terms of the log2 ratios that we use. In general, our normalized log2 ratios of greater than 1.5 will go to FISH confirmation. But our pathologists, our molecular pathologists, actually make those decisions. And I can tell you our group has definitely seen cases where the log2 ratio was less than 1, but clearly above the background of the rest, and those confirmed as bona fide amplifications by FISH. Those are especially problematic in low input materials. Our confirmation rate using FISH is very high. So if we find something suspicious by AMP and we do FISH, over 90% of those I would say confirm as bona fide amplifications. Where we run into problems, because we're now looking at really comprehensive whole genome coverage, is we run into polysomy, chromosome 7 polysomy being the most common for EGFR and MET, probably our most common confounder that would, in my opinion, should not lead to any treatment implications. So FISH confirmation, at least with the size panel we have now, is probably important for at least a subset of low-level amplification cases. True bona fide low-level amplification, and for those of you who run clinical labs and do HER2 FISH, I'm talking about ratios near 2.0, in my opinion, remain an area for more R&D. We've not been able to tackle this and really get our arms around it. CNV analysis, when you grind and analyze, is obviously majorly affected by the amount of contaminating normal stromal or normal other cells that are present and affect the denominator and numerator of your ratio analysis. So as you get down to lower and lower levels of amplification, which are clinically meaningful, i.e. HER2 ratios near 2, I think we still have a lot more work to do. This is the last one is my personal statement. I think it's a statement of fact, but I'll leave everyone out there to make their own decision. To me, FISH remains superior and will likely always remain the gold standard for copy number. But since it is so limited in its ability to multiplex, AMP or other NGS techniques clearly have advantages in cost and efficiency, and that's a compromise that I think we have to make. We may not be able to make that for HER2 and breast, but going forward, I think we will have to and should make that for most other tumors. Let's move on to rearrangements. We have most of our data and most of the excitement around the use of AMP and the Archer technology for detecting rearrangements. There are other ways to detect rearrangements. There's IHC. We'll talk about the expression levels of different rearrangements. Some genes are great. ALK is great. You can use immunohistochemistry to detect ALK. In fact, there's some great approved assays out there for ALK detection. ALK is not normally expressed in the lung, so for ALK positive tumors, IHC is great. ROS, not so great. We'll show you some data for that. Fish, I love fish. I've done tons of fish over the years. That was really important for the development of crizotinib. It's a reliable, good assay. It is technically challenging. You do need in-house expertise, pathologists, in my opinion, especially, and the ability to multiplex is very limited. RT-PCR is great. It works fantastic. You can design specific primers. However, you have to know both partners. And as we found out, and I'll show you in a few slides, that's really not practical going forward. The assay design for ROS1, I've really not outlined how AMP works in a multiplex manner, but it works very, very well in a multiplex manner. You can pull GSP1s into one tube, so you can put over 1,000 if you like, GSP1s into one tube, do PCR1, and then move that into the next tube, which is PCR2 with your GSP2s. And so you can multiplex the assay over 1,000. Excuse me. Talking too much. So by doing the reads and mapping them, you can map whether you're on the next exon, whether you map following this GSP 1 or 2 direction into the next fusion gene or into an intron. So as I said, we use total nucleic acid, so we will get all types of reads within any particular assay. In this particular case, we're looking for ROS fusion, so we're really looking to do 5' race in this case. So that's how the assay works. Why do we care about finding unknown partners? Because there are so many partners. As you know, for ALK, there's probably 20 or more, probably closer to 30 partners. Each of these 5' partners can have multiple breakpoints. RT-PCR becomes unwieldy, and you really are lacking clinical sensitivity. Clinical sensitivity. I'm going to skip over this slide in the interest of time. I think everyone's heard enough probably about fish. About the same time that we started working on ALK, the ROS rearrangements were described by the self-signaling group here up in Boston. Also a receptor kinase, similar structurally related to ALK in the kinase family. And as we found out later, also primarily in young non-smokers. So we began to screen for ROS patients by fish at the same time. ROS fish is a little bit easier than ALK fish because the genes generally are farther apart. And like ALK, the response of ROS-positive patients to crizotinib has been phenomenal. So you probably know this story quite well, but one of the important things to take home from all this is the prevalence is low. So many of the things we're talking about are very low prevalence. ROS is probably in the 1% range for lung, and to do 100 fish tests to find one ROS-positive case is quite expensive, quite tedious, and challenging. On the heels of ALK and ROS came RET fusions, obviously known in thyroid, then described in lung cancer, then NTRK fusions, then a Rolchinian's paper on FGFR fusions across a number of tumor types, including lung. And so it quickly became obvious to us that we needed to move quickly into the NGS space to look for multiple targetable kinases with the flexibility to look for novel fusions, novel fusion partners. This is a summary of our validation. So we developed a homebrew AMP assay. Long and Zhong Li developed this. Looking at ALK, ROS, and RET, we also added NTRK1 and PPR gamma for some of our thyroid cancer works, so kind of a lung thyroid panel. We looked at AMP performance versus fish, whether it's ALK fish, ROS fish, or RET fish, and AMP did extremely well in the clinical, what I would call the clinical validation. We detected almost all of the positives, and importantly, in some of the equivocal fish cases, and I'll show you one of those, AMP was able to solve some of our problems with unusual fish cases, either calling it a negative case for ALK there, or in this case, a ROS positive and a ROS negative one here. And so that really allowed us to resolve some challenging fish findings. So assay performed really well head-to-head versus fish, which we have a lot of data with. And that leads to a very important question around analytical sensitivity. I've had this discussion with many people here at MGH, within Archer, and with other people in the fusion space. How do you determine analytical sensitivity? It's actually, one slide does not do this justice. This could be, I wouldn't say a symposium, but at least an hour-long talk. The problem is standard serial dilution experiments are very challenging. And if you haven't thought about this, it's very easy to explain. RNA expression, unlike DNA, where you generally have 2 copies in a normal tissue, RNA expression can vary widely. So let's say you want to do an ALK, set up an ALK NGS assay, and you want to know what is your analytical sensitivity, what's your limit of detection. Okay, so you pull now several normal lung cancer cases or normal lung tissues, and you find your assay is quite sensitive. And why is that? Because ALK is not expressed in normal lung. So every time you add more RNA or cDNA, you're really not diluting out your target. If you were to take ROS, on the other hand, ROS is expressed in normal lung, and it is expressed fairly highly in about 30% of lung tumors. So now if you're normal, your quote normal expresses a fair amount of ROS, you're going to get a very, very different analytical sensitivity. So we were challenged for some time about how to approach this issue of sensitivity. And there's no easy answer. RNA expression varies significantly from normal sample to normal sample, from tumor to tumor, and it's not like DNA. From even if you have polysomy within a tumor, you might have 4 copies or 8 copies. You can talk several log difference of expression here, and that's a real challenge for a diagnostic lab to think about. Another approach, other than the standard serial dilution, I think the serial dilution is worth doing, in my opinion, to find a standard normal, let's say, or a small number of normals that you would use, and give a distribution of sensitivities. The other approach, which really would depend on having a large number of cases, the second 2 approaches depend on having a large number of positive samples, is you can get a large number of ALK or ROS or RET or NTRK, whatever fusion cases you're interested in, and look for statistical distributions of those. How many reads do you get for those positive cases? And if you had clinical follow-up, that would be even better. So that's something we're looking into, but you really need probably over 100 cases to be able to do that within any group. And the approach that we've taken has been to establish a minimal tumor percentage. So we found in our, I'm going to go back a few slides, we found in our validation set, you can see we have over 100. Between these, we probably have close to 100 positive cases. And so you can establish what is the lowest amount of tumor percentage that you've identified a true positive that correlated between FISH and NGS. And our cutoff currently stands around 15%. We think we can go lower for a certain target, especially ALK. We think we can go significantly lower. But this also depends on having a large number of cases. So this is an area that I'm happy to talk to anyone offline. I think it's a major challenge for the field. It's not an AMP-specific problem, but it's a question that I think most regulatory agencies really want to know, how are you going to approach. So I think we need to talk a little bit more about that. Skip quickly through this. This was our first 1,000 cases using this lung thyroid panel, about 6% positive cases, lots of RET, ALK and ROS, as you would expect, so about 6% positivity rate. On to the next slide. I'm going to skip over this in the interest of time, other than to say we can detect novel complex rearrangements. This was an indeterminate ROS FISH case. As I said, it's helped us solve some cases. In this case, we have a green-only dot, which we don't really know what to do with. In some of these cases, a green-only dot is on the 5' end of the ROS gene in our assay, and so in this case, the positivity was must have been a complex rearrangement which deleted the 3' probe, was confirmed positive by AMP. Even in the past year or so, there's been a number of papers, including this paper from the Blueprint Medicines group, that has described even more fusions, novel fusions of kinases in cancer. So we've worked hard to continue to keep up with the literature, which is not always easy. Within lung, for instance, the number of fusions has gone from ALK, RET, and ROS to probably we have about 20 genes now that we're actively looking at and need to keep track of. This paper came out of the Boris Bastian's group in 2014, just showing Spitz-Nevi melanomas, increasing number of fusions. So for this reasons, we've now moved to a larger 50-gene fusion assay, which we're excited about. We've launched and now use routinely. The first 150 cases of this assay, we were very excited that we had a significantly higher positivity rate, almost 10% of our cases. This was all tumor types. It's not limited to lung or thyroid. We found various fusions, and by fusions I include some RNA rearrangements such as exon 14 skipping and EGFR v3. I have just a few more slides. I'll try and finish up a little early so we can have questions. There's probably a lot of interest in the audience on exon 14 skipping. Do you look at RNA or DNA? We generally prefer using the fusion-based assay, the cDNA-based assay, to look for RNA. It gives you a very easy readout of no exon 14. But if you have DNA within the surrounding introns, you can find it. And this is a case that was positive for exon 14 skipping by RNA, where we have actually DNA genomic reads at the splice site of intron 14. And in a summary of 11 positive MET exon 14 skipping patients, we found almost all, well, in fact, all of them except for one we didn't have additional material, had DNA mutations that correlated with the RNA mutations. You probably know some of this data from other publications. This was limited largely to lung cancer, adenocarcinoma, and squamous cell carcinoma patients. And interestingly, almost all of them were never smokers. So along with ALK, ROS, another kind of non-smoke, and EGFR, another non-smoking lesion that's highly targetable. So obviously many people excited about MET exon 14 skipping. For expression, just in the last few minutes, you can look for RNA expression in this TNA-based assay, even if you're reading through into an intron. It's actually, you can use that data. To help correct for DNA reads. So in this case, we have basically our RNA coverage equals the read within the exon minus a very close intron coverage. So by subtracting this intronic coverage, you basically are subtracting almost all of the DNA, genomic DNA reads that are coming through this area. So that allows us to do profiling, expression profiling. This is a summary of our 50-gene fusion panel. These genes are on the bottom. The genes themselves are not really critical, other than to show you the data actually looks quite good using this normalization procedure that Song Lee has come up with. It's a log scale over here on the y-axis. And each of the genes, this is a summary of 200 different samples per gene, so this cloud is 200 different samples. And you can see widely variant expression from 0.1 up to 100, so you can have 3 logs or more of difference of expression using AMP-based expression profiling. In this case, this is ALK, right? This is a very low expressor, as we would expect in lung cancer. You don't see much ALK. And then you see some very high-level expressors, such as beta-2 microglobulin and other housekeeping genes here and here. So that brings up, just in the last minute or two, some very important issues. For expression, you have to balance primers. It's really important. And the reason is you pay a lot of money to sequence these high expressors. Unlike microarrays, where you might max out a particular spot on an array, you're not going to pay more money for that spot. If you keep sequencing your housekeeping genes, you're going to keep paying for sequencing. And I'm sure Illumina or Life loves that. You're just going to keep paying for sequencing. So you can balance the primers. You can bring down the primer concentrations as you design your targeted expression profiling assay to lower these levels. And that's, in fact, what we've done. These are normalized expression, but the raw read counts would be much lower than what we're showing you here. And we try and normalize them all so they're about the same. So that's really important, is to balance primers. The assay we're excited about, and we think it's working well. This is one example, a recent data set, again, 200 lung cancers, where we have ALK expression and ROS expression. ALK, you can see up here are mapped. Zhong Li has mapped the ALK fusion positive cases, which we know express a lot of ALK, versus the cloud of ALK negative cases. So we think we're really seeing true expression profiling. ROS, on the other hand, he's mapped the fusion positive ROS cases, and you can see those cases, the expression of ROS in the fusion positive cases falls right in the middle of the ROS cloud. And just for your edification, you can see that it's almost 2 logs difference median expression of ALK versus ROS. Here's beta-2 microglobulin as a comparison. So that's a quick run through expression. It's something that we're actively working on. I know the Archer guys are working on. So just to summarize quickly, AMP technology for targeted sequencing of RNA and DNA offers advantages of both hybrid capture and amplicon sequencing in one platform. You can do high confidence calls with complexity information from low quality, low input archive samples. The workflow can be scaled up to over 1,000 primer sets per tube, so you can do quite a bit of work in a single panel. I think, and I'm excited, at least in our R&D group, to continue the development of novel applications, panels, and basic bioinformatics and error correction tools. And then the last slide is just a thank you. This is a beautiful picture from Boston. No, I'm sorry, that's not Boston. But it's what I wish Boston would look like as we head into the fall and winter. Major thanks go to Long and Zhong Li from our team, who hopefully everyone can everyone still hear me? Okay. Just to thank Long and Zhong Li, who really did all the lion's share of work here. I'd like to thank also from the Archer team, Jason, Steve, and Josh, especially Jason, who founded Archer together with Long and myself, and it's been an incredible partner. We've learned a lot, not just about business, but about science. So I've taken up all of our time, but I'm more than happy to stay on for questions for those of you who can stay on.
Great, thanks, John, for that presentation. We do have a lot of questions, and due to time, we'll answer just a handful of them and address the remaining via email. So the first question: how do I best gauge the quality of my input material? Should I be using a fixed nanogram amount, or is there some other recommended way?
I think Archer has also adopted upfront. We've adopted some upfront QC steps. Archer also has adopted similar, I don't think identical, but similar upfront QC steps, which can save you a lot of effort downstream. For instance, for cDNA assessment, we have an upfront cDNA quality screen, which correlates really, really well to library success and sequencing success. So having those, I think, saves a lot. It's a little bit of work up front. It's a little bit of qPCR up front, but really saves a lot of time and effort. I think will make you and your techs very happy.
All right, great, thanks. Another question. For RNA from FFPE, what extraction method do you recommend? And are there any special tips to ensure that you get reliable data? Along with that, what is the average size of RNA molecules and the 5' 3' bias effect that you see?
I'm not sure I understood the second. I'm not sure I understood the second part of the question. Extraction of RNA, you know, we've tried a number of approaches, bead-based, spry cleanup-based approaches, as well as QIAGEN type column approaches. It's really, I don't like to recommend. I would leave that more to the Archer, your Archer team to comment on that. We found that multiple technologies work just fine for RNA. Doing the upfront QC, I think, will let you assess very quickly whether you're getting high enough quality RNA. With that said, I do think the major challenge people have had to adapting AMP technology has been poor RNA quality in their own laboratories from FFPE. So it's not that extraction is not the critical step. I think it is actually the most important step because the technology works, I think, fairly simply. I'm happy to talk offline about specific products, but I'd rather not recommend anything. What is the average size? What is the average size?
Ooh.
The second part of the question, what is the average size of RNA using AMP? Our sequencing uses 150 base pair paired reads. We don't really dictate. The FFPE material dictates the size of the RNAs. I would say it's anywhere between 100 and 300 base pairs, averaging 150 to 200. But that's not really at our discretion. It's more the condition of the RNA. But if you have it, you need probably about you need somewhere in the 100 to 200 base pairs to get enough reads that you can get. You need to map both to your gene of interest as well as a potential partner. So you probably need 20 base pairs on both sides plus your primer sequencing. So there are a minimal RNA length that is required for the assay, but it's generally not been a challenge for us.
Great, thanks for that answer, John. Another question, how about DNA-seq-based fusion detection instead of cDNA?
Good question. As I said, I didn't want to have an exhaustive review of other technologies. I think there's many other technologies that could have been looked at for fusion detection. The problem with genomic DNA, and I'm just going to kind of make it a very explicit answer, hopefully not oversimplifying it or insulting anyone, but most fusions are within introns, and some of those introns can be very long. So to do fusion detection from introns, you pay a lot of money. You have to, in many cases, tile across large, very large introns, and you pay for all of that sequencing. So for the same amount of money that you could put maybe 5 or 10 probes to look at, let's say, the alk kinase domain, and the amount of sequencing that it takes to read into through that cDNA, you may need 50 or more probes to tile, probably more, hundreds, to tile across those, and you pay for that sequencing, and that can be expensive.
Thanks, John. I'm glad that question was asked. Next one. What do you think about Dr. James Wiley's approach to using competitive template spike-ins to accurately quantify starting template for the targeted RNA and DNA-seq applications?
I like the idea. I think artificial templates, artificial spike-ins, all of those are really valuable, and I think are a good step. I didn't include that as a possible step or approach to the analytical sensitivity. Artificial templates, I think, do have an important role. I do like to be very explicit, however, that we like to think as molecular diagnosticians that we can predict the sensitivity in every sample that is in front of us. And what I'm trying to point out is the RNA expression of any of your targets. Let's say you have a 50 gene panel. Those 50 genes in the surrounding normal lung tissue can be highly variable. And so although your spike-ins can give you some assessment of how the assay itself performs, it doesn't give you complete confidence in every single sample that's in front of you because of the biology. It's not the technical limitation, it's the biological limitation that we have to come to terms with. So we can figure out, yeah, this assay performs as well as any other assay out there on the market, and you can use spike-ins to do that. It doesn't help me as a clinician saying, in the patient in front of me, we have a 5% sensitivity, when we really don't know because the RNAs are so widely expressed. I hope that made some sense.
Great, thanks, John. We just for the sake of time, I think we're going to close, and we will we'll answer the remaining questions on a one-on-one basis via email. We'll follow up with you. Today's webcast will be available for on-demand viewing both on labroots.com and on archerdx.com, and we'll let you know when that's available, so you can revisit or you can share it with people who weren't able to come. Thanks to Dr. Iafrate for taking the time to prepare and present your work, John. Appreciate it. And also thank you all for attending this webinar. And we'll see you next time. Have a good day.